Assalamu alaikum and welcome everyone to the second part of our molecular biology preparation series, Molecular Techniques. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experience with all of you through this series. So let's dive in together. In our previous lecture, we explored various PCR variations. Today, we will cover the final segment of nucleic acid amplification, other nucleic acid amplification techniques. As the molecular diagnostic continue to evolve, alternative nucleic acid amplification techniques have been developed to address different diagnostic challenges. Unlike traditional PCR, many of these methods operate under isothermal conditions, eliminating the need for complex thermal cycling while maintaining high sensitivity and specificity. Today, we will explore several key techniques that are widely used in clinical laboratories, such as the hybrid capture, nucleic acid sequencing based amplification. And we will talk about transcription mediated amplification and loop mediated isothermal amplification in next lecture. So, each of these methods offer unique benefit, enhancing the speed, efficiency, and accuracy of molecular testing. Let's explore their mechanism and application in the greater details. So, welcome to the session.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.
All right, now let's move to the question section and I will start with hybrid capture. All right, now let's see the first question we have today in hybrid capture. So the first question here, which of the following best describe the hybrid capture assay format? The correct answer is capture hybridization assay. Actually, hybrid capture assays work by hybridizing target nucleic acid with the specific probes. Once bound, they are detected using antibodies, making the process highly sensitive for molecular diagnostic. Let's move on. All right, the next question here. Hybrid capture detection primarily relies on which one of the following? The correct answer is signal amplification using antibodies. Actually, hybrid capture uses labeled antibodies that bind to hybridized probe target complex, producing detectable signals. All right, let's move on. All right, next question here. How does signal amplification occur in hybrid capture assays? The correct answer via enzymatic reaction producing chemiluminance. All right. Actually, in hybrid capture assays, signal amplification occurs through the enzymatic reaction that generates chemiluminance signals detectable by specialized instruments. All right. Let's move on. Okay, the next question here. What advantage does hybrid capture have over PCR-based methods? The correct answer is less prone to contamination due to the lack of amplification step. As we know, hybrid capture doesn't involve amplification of nucleic acids, so reducing the risk of contamination compared to PCR-based methods. All right. All right, the next question here. Which enzyme is commonly used in the detection step of hybrid capture assays? All right, the enzyme alkaline phosphatase is commonly used in these methods as it produces a chemoluminescent signal upon binding to DNA RNA hybrids. All right, let's move on. Okay, next question here. What is the role of the capture probe in hybrid capture assays? Actually, the capture probe in hybrid capture assays is designed to hybridize specifically with the target nucleic acid, allowing for its detection. All right, let's move on. All right, in hybrid capture assays, what type of nucleic acid is used at the probe to detect viral DNA? Actually, hybrid capture uses single-strand RNA probes to bind specifically to viral DNA targets. Okay, let's move on. Okay, the question here, what type of probe is used in hybrid capture assays when detecting viral RNA? We should differentiate between this question and the previous one, all right? All right, the correct answer is complementary DNA probes. Actually, hybrid capture technology detects viral RNA by using complementary DNA probes that hybridize with the RNA targets. This hybridization form a probe target complex, which is then detected through signal amplification, producing a measurable output. Okay, let's move on. All right, in hybrid capture it says, what is the purpose of the hybridization buffer? All right, the correct answer here is to stabilize the DNA RNA hybrids. All right, let's move on. In hybrid capture assay, what is the significance of using high affinity antibody? The correct answer is to enhance the specificity of target detection. All right. Actually, high affinity antibodies are used to specifically bind to DNA RNA hybrids, ensuring accurate detection of the target nucleic acids. All right. Let's move on. All right. Next question here. What is the role of chemoluminescent substrate in hybrid capture assays? All right, the correct answer is to produce a visible light signal upon enzymatic reaction. Actually, the chemoluminescence substrate react with the enzyme. Which enzyme? Alkaline phosphatase, to produce a light signal that can be measured. All right, let's move on. Okay, the next question here. Which of the following is not required in hybrid capture assay? The correct answer is RNA polymerase. Actually, hybrid capture doesn't involve RNA polymerase because it doesn't amplify nucleic acids like PCR, all right? Okay, the next question here. Hybrid capture is particularly useful for detecting. The correct answer is early stage viral infection. All right. Actually, hybrid capture detects low levels of viral DNA or RNA, making it ideal for early diagnostic of infection like human papilloma virus and hepatitis B virus. All right. Okay. Which of the following is the key advantage of using hybrid capture over traditional culture methods? The correct answer is faster turnaround time for result. Actually, hybrid capture says provide faster result compared to the traditional culture method, which can take several days to weeks. Alright, the next question here. A major disadvantage of hybrid culture compared to PCR is it cannot quantify viral load. Actually, hybrid culture only detects the presence of viral genetic material, but it doesn't provide quantitative result, unlike PCR. Okay? Alright. What is the main application of hybrid capture in clinical diagnostics? The correct answer is monitoring of viral load in patients. Alright, let's move on. Okay, here is the last question for hybrid capture techniques. Which clinical test commonly use hybrid capture technology? The correct answer is HPV screening. Actually, hybrid capture is widely used for HPV screening, helping detect high-risk human papillomavirus infections that may lead to cervical cancer. All right, let's move on. If you found this content helpful, don't forget to like, share, and subscribe. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. Okay, next we explore nucleic acid sequence-based amplification, NASBA. All right, let's see the first question we have here. The first question is, which of the following is the key advantage of NASBA over traditional PCR? The correct answer is ability to amplify RNA directly. Actually, NASPA is the RNA-based amplification technique, can amplify RNA directly without the need for a DNA intermediate, making it particularly useful for detecting RNA viruses and other RNA targets. All right, let's move on. Okay, which of the following is the primary enzyme used in NASPA to amplify RNA? The correct answer is RNA polymerase. Let's move on. All right, next question here. What enzyme plays a central role in NASP reactions? The correct answer is reverse transcriptase. Actually, NASBA techniques uses reverse transcriptase to convert RNA into cDNA, which is then amplified, making it highly effective for RNA virus detection. Okay, let's move on. In NASBA, which of the following steps is not involved in the amplification process? The correct answer is denaturation. Actually, NASPA doesn't require a denaturation step because why? It amplifies RNA directly at a constant temperature, unlike PCR, which requires thermal cycling. Okay, let's move on. Okay, this question here. What is the typical temperature range for NASPA reactions? Alright, actually, NASBA reactions are typically carried out at a constant temperature of 37 to 42 degree, which is suitable for enzyme involved in the process. Okay? Alright, which of the following is the common application of NASBA? Correct answer is detection of RNA viruses. As we know, NASBA is commonly used for detection of RNA viruses because it can directly amplify RNA, making it powerful tools for diagnostic viral infections. All right, let's move on. In NASBA, what is the function of the T7 RNA polymerase? The correct answer is to synthesize RNA from a DNA template. As we know, T7 RNA polymerase synthesizes RNA from a DNA template, which is the crucial step in NASPA process to generate multiple RNA copies. Okay? Okay. Which of the following best describes the amplification mechanism of NASPA? The correct answer is exponential amplification of RNA. As we know, NASPA achieves exponential amplification of RNA through a series of enzymatic reactions, making it highly efficient for RNA detection. All right, the next question here. NASPA differs from PCR because the correct answer, it operates under isothermal condition. Actually, NASPA is the isothermal process, meaning it works at a steady temperature, making the process much simpler, unlike PCR, which constantly shifted between heating and cooling cycles. All right, let's move on. All right, this question here. NASPA utilizes a unique mechanism involving. All right, the correct answer is RNA to RNA amplification. Actually, NASPA enables direct RNA to RNA amplification, bypassing the need for DNA intermediates. Unlike traditional methods, NASPA specializes in detecting RNA viruses like HIV by efficiently generating RNA copies under isothermal conditions. All right, let's move on. All right, this question here. What distinguishes NASPA from RT-PCR in RNA amplification? The correct answer is NASBA performs amplification at a constant temperature, unlike RT-PCR, which requires thermal cycling. All right, let's move on. Okay, this question here. NASBA relies on a combination of which three enzymes for RNA amplification? The correct answer is reverse transcriptase, RNase H, RNA polymerase. Actually, NASBA uses reverse transcriptase to generate cDNA, RNase H to degrade RNA strands, and RNA polymerase to amplify the new RNA copies. All right? That's all for today. I hope you enjoyed this lecture and look forward to hearing from you what you think of it. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. And don't forget to click here to watch the previous videos. Salam.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.